Hi kids, good morning. Welcome to Anatomy and Physiology Laboratory. I am Sir Winbrand Corpus and I'll be your lecturer for today. So our topic for today is about cellular permeability. So before we start, we'll first define what permeability or permeable means. The term permeable means that the structure or a cell permits the passage of substances through it, while the opposite of permeability, which is impermeable, means that the structure does not permit the passage of substances through it. So we have the following objectives. We could first define what are hypotonic, isotonic, and hypertonic solution. We will be able to discuss its effect on the red blood cells or other cells and apply the knowledge about tonicity. So I will be challenging you to answer the following two cases. So if you have answers or you are willing to share your answers with me, I am very willing to accept your answers. Please send your answers to my Telegram account. So case number one. Brian, 15-year-old male, is performing an experiment about red blood cell tonicity. He was instructed by his preceptor to put 2% sodium chloride in the RBC suspension. What do you think will happen in the red blood cells? Will they crenate or will they be hemolyzed? And why? Case number two. Hannah, 11-year-old, presents with cough, fever, and palpitation. Upon physical examination or PE, she seems dehydrated and with sunken eyeballs. The attending physician ordered to give D5 water. So D5 water is the intravenous fluid that was given to her. What tonicity of the IV fluid did the doctor give to maintain her intravascular volume? Is it isotonic, hypotonic, or hypertonic? And what would be the expected morphology or blood picture of the red blood cells? Okay, so we will also define the term osmosis, which is the movement of water or the net movement of water molecules through a selectively permeable membrane. Next, we have osmotic pressure, in which it is the pressure exerted by the impermeable solutes, for example, the proteins, example of which is albumin, in which if there is higher solute concentration, there would be a higher osmotic pressure. Next would be tonicity. It is the measure of the solution's ability to change the volume of cells by altering their water content. So we have discussed that we have three types of solution. We have an isotonic solution, a hypotonic solution, and a hypertonic solution. So we will be first discussing what is an isotonic solution. So isotonic solution is any solution in which the cell, for example, in this case, we have the red blood cell, maintains its normal shape and volume. So that defines an isotonic solution. They say, or some journals would say that they have the same salt concentration as the normal cells of the body and the blood. So they also have equal osmotic pressure. For example, 0.9% sodium chloride or the plain normal saline solution. Next, we have Ringer's lactate and D5 water. So in an isotonic solution, the concentration of solutes that cannot cross the plasma membrane are the same on both sides of the membrane in this solution, in isotonic solution. For example, 0.9% NaCl yung ginamit natin or the PNSS. So, kaya siya tinawag na normal physiologic saline solution kasi isotonic siya. And then, yung mga RBC natin, it will retain its normal shape. So, kung, for example, biconcave yung shape niya, so it will remain biconcave in an isotonic solution. Because the RBC plasma membrane permits the water to move back and forth. For example, kung 100 ang naglabas, 100 din yung papasok. And then, since i-permit niya yung passage of water back and forth, but it behaves as an impermeable to sodium and chloride or yung mga other solutes. So yung water lang yung nakakalabas. So meaning isotonic solution siya. So this would be the blood picture of the normal red blood cells in an isotonic solution. So ang RBC dito is retaining their normal shape and volume. Yan. Next, we have a hypotonic solution. So, a hypotonic solution is different from the isotonic solution. Paano? So, hypotonic solution has a lower osmotic pressure than that of the body fluid. And then there is a water movement into the cell. Kanina sa isotonic solution, meron daw siyang equal osmotic pressure. And then the water movement is equal. So kung 100 yung maglabas, 100 din yung magpasok. But in hypotonic solution, there is the water movement into a cell. Again, there is water movement into the cell daw. Papasok daw yung water. So in this case, the water molecules enter the cells faster than they leave. For example, 100 yung nagapasok sa cell, pero yung nagalabas lang is 25%. So this would lead to swelling. So maging swollen yung RBCs natin. From the normal shape, maghabok siya, swollen, or mag-burst. That would lead to hemolysis. Kaya dito, mapansin nyo, I have this mnemonic, hypotonic. Hypo, O-O-O, leads to swollen or O-swollen, and bursting or bursting. That would lead to hemolysis. So, O-O-O, hypotonic, swollen, bursting, and hemolysis. So, hemolysis, by the way, is the rupture or magputok yung RBC, maglyse. And then, example of the hypotonic solution are pure water, 0.45 sodium chloride. So, as you can see in the picture, yung water goes into the cell. Yung kanina sinabi ko, if 100 yung pumapasok na water, tapos 25 lang yung lumalabas, so the net result would be the swelling of the RBC and eventually will lead to the rupture or bursting or hemolysis of the RBC. So ito na yon. So for example, ito, malaking RBC na ito siya, naging swollen na daw siya kasi tingnan niyo yung central pallor, lumiit. So swollen na daw siya. So pag nag-swollen siya, it has the tendency to burst. Tingnan nyo ito, parang nagputok na siya ito din. Putok, nagputok na yung RBC. So next, hypertonic solution. So they say that in hypertonic solution, there is higher osmotic pressure than that of the body fluid. Meaning, mas marami yung amount ng solute sa labas ng red blood cell or sa environment ng outside the red blood cell. So, ang tendency is, yung water pupunta doon sa labas ng RBC. So, there would be water movement out of the cell. When we compare this to hypotonic solution in which ang concentration daw ng solute dito sa labas, itong sa blue, is mas mababa kaysa sa concentration na nasa loob ng RBC. For example, 100% solute ang nandito, tas dito 25%. So ang tendency is, yung water daw is papasok sa cell. Yun yung sa hypotonic solution. While in the hypertonic solution, dito naman daw yung 100, sa labas daw yung 100 dito sa blue, tapos dito 25. So, 'di ba, ang principle ng water is kung saan maraming solute, kailangan nagpunta doon yung water para i-dilute niya. So 100% daw yung 100 solutes daw nandito sa labas na nasa color blue. And then 25 inside the red blood cell. So the water now moves out of the cell. So when the water or the solvent or the water na lang in this case will move out from the cell, it will lead to shrinkage and crenation of the red blood cell, meaning shrink, magliit siya. As you can see, nagliit siya. And then it is crenated, meaning parang na-crumple na siya or para siyang sea urchin. Kasi nagliit naman siya. So, mnemonics, again, hyper, hyper, R, shrink, and crenate. So, ito na. This would be the blood picture of a crenated RBC because the water inside the RBC came out or naglabas from the RBC to the outside environment. Yan na yun sila, mga crenated or shrink RBC. So, this would be the summary of the movement of water in isotonic, hypotonic, and hypertonic. So, first picture is hypotonic. Tingnan nyo, the water molecules goes into the cell. So, meaning papunta sila sa cell, it would lead to O, hypotonic, swelling or swollen RBC, and then bursting, and eventually hemolysis. In the middle picture, it is an isotonic solution. As you can see, if may nagpasok na dalawang water molecule, may maglabas din na two water molecules. So there is an equal movement of water molecules. Next would be the hypertonic solution, the third picture here. So as you can see, tingnan nyo, the movement of water is from to the outside of the cell. So there is movement of water papunta sa labas. So ito, tingnan nyo, the water comes out or maglabas siya papunta sa environment. So the result would be hypertonic. So there would be shrinkage and crenation of the red blood cells. So ito, basically, the same principle. So in hypotonic, magpasok yung water and then they will be hemolyzed, magputok. In isotonic solution, water in, water out. In hypertonic solution, water out. So, palabas yung water. So, kung ang hypotonic solution, swollen, ang hypertonic solution, shrink. So, be familiarized sa blood picture because pwede natin to ipalabas sa exam na ganito lang. So, in hypotonic solution, it has swollen RBC, it has a tendency to hemolyze or burst. In isotonic solution, there would be normal shape and volume of the red blood cell, meaning isomorphic sila, equal yung morphologies nila. And then in the hypertonic solution, the red blood cell would appear crenated or they will shrink. So please answer the following cases on your own, and if you're willing to answer it and send it to me, so please do it. So for the references, we have Tutorial Principles of Anatomy and Physiology, pages 62 to 63, and also the SPC Laboratory Manual. So any questions? And thank you for listening. Study well, kids.